Now, in this part of the chapter, we'll talk about the classification of the cells of the nervous system. Because there are different functions, remember we said we have a sensory function, motor, and integrative function. The nerves, we do have also 3 kinds of cells according to their structure. So the kind of cell that we talked about is the multipolar cell, and that makes about 99% of neurons. Those make up your motor neurons and integrative neurons. They have multiple dendrites, and that's where the name multipolar comes from. And as usual, they have that one axon. The next kind of neurons that I want to introduce are these bipolar neurons. These are for special senses. So they will carry senses that are only found in the head and neck. For example, vision. Now, for example, these are all of the special senses we have. Vision, smell, taste, hear, and equilibrium. So all of those are considered special senses, and they are carried by these bipolar neurons that have this very one long dendrite and a very long one axon. Okay, so they have 2 processes, hence the name bipolar. Now this unipolar neuron, you can see here it has that one process and then divides into 2 processes. A peripheral process that goes towards the peripheral nervous system, and a central process that will go towards the brain or the spinal cord, so towards your CNS. These are for general senses, okay? And then a collection of, all of these cell bodies together. So obviously each nerve is not made out of just one axon. It's made out of multiple axons. Now, these multiple axons are going to have multiple cell bodies. When these cell bodies collect together, they make a ganglion. Ganglion is single, ganglia would be plural. So cell bodies collected together in the peripheral nervous system is known as the ganglia. Now, we've talked about the classification, and now let's kind of tie all of this information together. And hopefully at this point, we're all comfortable with what we're talking about here. So this part represents the central nervous system, so brain and spinal cord, while this portion represents the peripheral nervous system. We have sensory receptors, and those are very specialized nerve endings that detect changes in the environment. Remember, these changes can either be external changes or internal changes. Now, these receptors pick up those changes, and the changes will propagate on these sensory neurons, also known as the afferent neuron, into the CNS. It will be integrated. A lot of data will be collected, integrated, and decided upon what are we going to do about these changes through these inner neurons. Notice that inner neurons are present only in the CNS. They do not have any extensions out of the CNS. So inner neurons are found exclusively in the CNS. Now the inner neuron, it'll integrate, make decisions, and these decisions will be carried on a motor neuron, also known as the efferent, and it will take those motor orders to the effectors, again, on your efferent neuron. And these effectors, again, are either muscles or glands. A way to remember which one is afferent and which one is efferent would be through this word here, same. So sensory afferent with an A, motor is efferent with an E. Okay, so remember that your sensory neurons take towards the CNS, and those could be either unipolar if they were general senses, or bipolar neurons if they were special senses. Inner neurons and motor neurons, those are both multipolar. Now, what about the neuroglia? So we said neuroglia are your helpers, supporters of the neuron. They actually support the neuron even, since literally before birth. So in the embryo, it'll guide the neurons where they would end up going. So the nerves know that, okay, I'm a nerve of the lower limb because of, thanks to the help of these neuroglial cells. Okay, the neuroglial cells also make growth factors and that help to nourish and grow the neuron. It will remove any excess ions. So when we talk about how the action potential propagates or spreads on the nerve, we are going to be talking about a lot of ions, in particularly sodium and potassium. So these ions these neuroglia are responsible in removing any excess ions that might, mess up with this whole action potential. It also helps in the making of neurotransmitters or the removal of excess neurotransmitters. It aids in the formation of a synapse. So remember, a synapse is where a nerve meets something else. So it could either meet another nerve or a muscle or a gland. So the neuroglia in the CNS. So pay attention that these neuroglia that we're going to be talking about are in the central nervous system. We have astrocytes, and I'm going to go to the next image to explain them, but let's introduce them first. Astrocytes, and they look like a star, hence the name astrocyte. Oligodendrocyte, microglia, ependymal cells, and let's take a look to introduce what they look like and what they do. So we'll start off with this astrocyte, and you can see here that it has multiple extensions. One side of the extension goes to the neuron, while the other side would go to the capillaries. Remember, these are in your central nervous system, so this could either be in the brain or in the spinal cord. There is no direct contact between nerve and blood. So the astrocyte helps in making what is known as a blood-brain barrier. So anything that needs to go from blood to neuron would have to go through these basically suction cups, go through the astrocyte, and then to the neuron. So if you're giving a patient a medication and you want that medicine to affect the brain or spinal cord, well, it would have to be a kind of medication or a kind of drug that would cross the blood-brain barrier, because if it doesn't, it will have no effect on your neurons in the CNS. Okay, so again, astrocytes help in the transformation of also nourishment, so oxygen and any glucose these neurons might need, and it gets rid of the waste products like carbon dioxide to go back to the capillaries. So those are your astrocytes. Next off, we'll talk about these snowflakes that we call microglial cells. Micro means small. They are specialized macrophages. So remember, macrophages are part of the immune system. They eat up germs and pathogens or any bug trying to invade the body. Well, these cells are changed and modified in the CNS, and we give them a special name known as the microglial cells. So these are basically parts of the immune system. They are going to, again, look around and get rid of pathogens. Now the oligodendrocytes, so let's break down the names. Cyte means cell, dendro means dendrites, and oligo means few. So these are the cells that have few dendrites. And you can see here that because of these few dendrites, they are helping in the making of the myelin sheath in the CNS. There's one last cell to talk about, and these are the ependymal cells. These cells line the cavities of the brain and spinal cord. So the cavities in the brain are known as ventricles, while the cavity in the spinal cord is known as the central canal. These cavities are full of cerebrospinal fluid, or CSF. That fluid is made and maintained by the ependymal cells. Now for the neuroglia in the peripheral nervous system. Okay, so they're different. We've got Schwann cells. Schwann cells make myelin sheath in the peripheral nervous system. And remember, we said that it speeds up the transmission of action potentials. We also have satellite cells, and satellite cells support those little clusters of cell bodies that we called ganglia. So make sure you are comfortable with the fact that myelin sheath in the CNS are made by these oligodendrocytes, while myelin sheath in the peripheral nervous system is made by Schwann cells. Now, what about neuroglia and axonal regeneration? So if an axon or if a nerve cell is damaged, will it regenerate or not? Well, that really depends on, is the nerve part of the peripheral nervous system or the central nervous system? So if it's in the peripheral nervous system and the cell body is intact, that axon can regenerate. Okay, so if a peripheral axon is injured, but the cell body is intact, that can lead to regeneration. And I'll show you in the next slide how that happens. But if the injury is in the CNS, okay, that whether the cell body is injured or not, regeneration does not occur in nerves in the CNS. And that's why if somebody gets a stroke that would lead to, the complete death of certain neurons, it is almost impossible for them to regain complete function. Okay, so any neurons we lose in the CNS are pretty much lost forever. Okay, so regeneration is very unlikely when it comes to neuron damage in the CNS. So why is that? Let's take a look at what happens in the peripheral nervous system so that we can understand why we do not regenerate in the CNS. So right here is your cell body, and you can see the axon, and there is your skeletal muscle fiber. So what happens is that you can see the injury. This is the site of the injury. Remember that oxygen and all of the neurotransmitters and everything would be going down the axon to take those needed, goodies throughout down the whole, the rest of the axon. So if you're injured here, this part peripheral to or proximal to the injury can survive. But the parts that are distal to the injury are going to die off because they're not getting their oxygen supply. Know that these Schwann cells are individual cells, so their oxygen supply and nutrients is totally independent of the cell body. So these Schwann cells are going to survive, those remain, and it's because of the Schwann cells. Now they survive, they make a tunnel, there's a guiding tube where this axon can regenerate, and it knows where to go because, thanks to these Schwann cells. So the neuron regeneration in the peripheral nervous system really depended on the survival of the Schwann cell, the survival of the myelin sheath, making a tunnel or a guide as to where the axon can regenerate and reach its destination and try to maintain those connections or to reestablish those connections. It might not always be perfect, but it's definitely better than nothing. In cases like this, you want to tell the patient that it's a very slow process. So the growth of the axon is about 1 millimeter per day, which is extremely slow. So if somebody has an injury, say in the groin where they injured a nerve, it could take up to years for them to regenerate that whole axon, and they could feel that numbness, in the heels for years and years, which could be very frustrating for the patient. So knowing on a scientific level that, it is regenerating, you just have to wait. It's not easy to tell that to patients, and again, something you might have to explain to them, although it is still frustrating for them to hear that. But on the other hand, in the central nervous system, myelin sheath, for unknown reasons, do not survive. Okay, so remember the myelin sheath in the CNS are made by oligodendrocytes. Those cells do not survive if neurons are injured, and that is why there is no neuronal regeneration when it comes to the CNS neurons. So this ends this part. I will see you in part 3.